Topic B2, I'm going to do this in 2 bits. The first bit's about proteins. And proteins have a few different functions. Very important for structure. If you think of your hair and nails, things like keratin, collagen. Enzymes consist of proteins. An example of that would be amylase. They also constitute hormones, which are these chemical messengers in the body. An example of that, insulin. And they also have transporter roles. For example, hemoglobin moving the oxygen around our bodies. So the general structure, we start with an amino acid here, and we can see we've got a carbon with 4 groups attached to it, one of which is a carboxyl group. We've got an amino group, hence the name amino acid, and all of them have this H, and then of our 20 different naturally occurring amino acids, they all have a different R group, and it could be something as simple as H, you can see here, or it could be a group like that. So depending on what that R group is, affects the name of the amino acid, and it's obviously going to affect the properties of the protein ultimately. So these amino acids join up by condensation reactions, and you can see that I'm going to join the carboxyl group with the amine group there, and I form what's called a peptide bond. If that continues, I end up with a longer chain. Now, amino acids are referred to as zwitterions. Zwitterions have positive and negative charges on the molecule, but overall their net charge is 0. Amino acids are amphoteric because they can react with both acids and bases, and they're also amphiprotic because they're able to accept or lose an H+. If you think of this, this could be lost or an H+ could be gained there. So depending on what kind of solution they're in will affect their structure. So if we're in acidic solution, there's lots and lots of H+'s around, then we're going to stick an H+ on the carboxyl group. If we're in an alkali solution, there's not many H+'s, this is going to tend to deprotonate, so you can see we've lost it, our H+ from there. So the amino acids also have a buffering ability. So this idea of gaining and losing protons from our amino acid leads to an isoelectric point. So at our isoelectric point, you can see we've got our zwitterion existing, and above or below that we're going to expect to see maybe an overall charge on our amino acid. So at these isoelectric points, this is what the amino acids look like. And that idea is important in enabling us to separate amino acids. So if we were to put these amino acids in a buffer of a certain pH, some would maybe be protonated, some would maybe deprotonate, and ultimately we'd end up with different amino acids carrying different charges. So when we put a potential difference across that gel, some aren't going to move at all if they don't have a charge. For example, if they're at their isoelectric point. Others are going to shift towards the anode, others are going to shift towards the cathode. So if I think of an amino acid and I put my amino acid in a pH that's higher than its isoelectric point, in other words it's quite alkaline, then I'm going to find that I'm going to have a negative charge. And that's going to then be attracted towards a positive electrode, the anode. So let's say the pH was 12, for example, I might end up with something like this happening. If I put it in something that's quite acidic, if my solution is maybe at pH 2, lots and lots of protons, I'm going to be protonating both parts of that amino acid, like that. You can see now it's positively charged, and this is going to move towards a negative cathode, so they're going to split up. And we have to consider as well that our functional groups can also play a part, whether these are going to be protonated or deprotonated too. And that affects their isoelectric point. So another way of doing it, so I should say that the first step in this process is hydrolyzing your peptide. So if you've got several amino acids joined together and you want to see which amino acids are present, you've got to hydrolyze it. You can hydrolyze it either using enzymes or 6 molar hydrochloric acid. You could, if you wish, if you didn't want to do the electrolysis, you could run that simply as a chromatogram, and what that involves is dotting it on some sort of surface, maybe just chromatography paper in the lab, running a solvent up that, so you've got your dot there, making sure it's above the line of the solvent. Solvent starts to move up the page, and depending on how strongly this binds to the paper or the solvent, different amino acids will move different amounts, and you'll see them split up. And we can mark off where those points are, and that will give us for each of those dots or amino acids a retention factor. And you can see there how the retention factor is worked out, and that retention factor for a particular solvent will be characteristic of that amino acid, so you can see which amino acids that you have. So for example here, the retention factor of that would be 3.6 divided by the distance moved by the solvent, which is 6.0, and then you could look up that value in a data booklet. If we wanted to go the electrophoresis route, then again we still need to separate them. We've got to do that hydrolysis step, and you can see here typical setup that might be used, and our amino acids, depending on their charge, maybe they've got no charge under these conditions, but if they're positively charged, they're going to be heading that way. If they're negatively charged, they're going to be heading that way. And by looking at which way they're moving, you can start to deduce which amino acid you're working with. I should have highlighted back here that the last step in looking at this paper chromatogram is spraying it with some sort of agent such as ninhydrin, so that you can actually expose where these seemingly colorless amino acids are. So here's an example of processing the results of electrophoresis gel, and you can see it's been run at different pHs, and so you're looking to see maybe, well, who's not moving, because if they're not moving, then overall they're not going to be charged. So if you look at pH 6, you can choose one from there, and then thinking about, well, which way are they moving? And there's a little bit complication here in that there's no positive and negative on the diagrams, but you can see they're moving in opposite directions, and over here one of them is moving even more, so that might imply that it's got a greater charge to it. And if you look down there in the box, you'll see the solution, so you can work through that and have a look at it. So 4 types of structure that we need to refer to when we're talking about amino acids. Primary structure is very simply the sequence of the amino acids. So just what have we got joined up? Glycine, alanine, leucine, etc. And you see this long chain. That's our primary structure. Secondary is where we're going to see some additional interactions between amino acids. So we end up with often regular repeating patterns, such as the alpha helix, where you can see this hydrogen bond between residues that are 3 or 4 places apart, and it gives this helical structure. Another secondary structure, such as silk, is a beta pleated sheet. Again, you can see maybe this is going to give the protein different properties. And then longer range and kind of more diverse intermolecular forces will lead to a tertiary structure. So here's our secondary structure, just some local folding like a helix. But then when those helices start to interact with each other for further distances around the chain, maybe across that type of area like that, we end up with these unique shapes, and that's where we end up with sort of enzyme active sites and so on. And so these intermolecular forces could be things like hydrogen bonding. They could be salt bridges, where we've got charged parts of the molecule. They could be hydrophobic side chains, so certain groups, maybe like benzene rings, will want to group together and form London dispersion forces, so they'll fold on top of each other. And then if we have 2 cysteine residues, so cysteine has that, if we get 2 of them, we can get this disulfide bridge, so we actually get a bond between these 2 sulfurs linking those amino acids. And you can see here in the diagram some examples of those types of interaction that are forcing our protein to bend and bond into a particular shape. And that shape, as I mentioned, is important in leading to these very specific active sites. So here's some examples of tertiary structures. You see these myoglobin, cytochrome P450BM3. They're quite intricate, complicated foldings. And then beyond that, where tertiary structures interact, we can make quaternary structures. And probably the example that you'll come across here is hemoglobin, where we've got these 4 subunits that are held together by various interactions, and we get this much larger molecule. So just comparing different types of protein, whether they're globular or fibrous. So examples of fibrous proteins, keratin, collagen, are usually structural. Globular stuff, more like enzymes, quite soluble for the globulars. As the name suggests, they're sort of compact, spherical. Whereas with these fibrous ones, they're longer chains and they're generally insoluble. When we talk about these structures, obviously the structure is vital to the function that the protein or enzyme, as it probably is, performs. We can change the shape, in other words, denature what we've got, this irreversible change, maybe by exposing it to a very high temperature. That's going to affect the hydrogen bonds. If we expose it to strong acids, strong bases, that's also going to affect the hydrogen bonding. Putting it in some concentrated salt solution is going to start to disrupt the salt bridges. Organic solvents might affect the hydrophobic interactions, and detergents will also have the same effect. So these are ways that we can destroy those structures and as a result denature our protein or enzyme.